On to the last part of the morning's program, which is talked by George Whitesides. When I think of somebody to tell us about innovative ideas in nanoscience and how they've been useful in a variety of fields, especially including biological fields, I can't think of anybody who could give a more appropriate talk. So,
but I'm not going to talk about any of that.
well,
I'm not a neuroscientist, but what I have done successfully survived for a long time in science. And so I want to give you some kind of sense of a little bit about how this effort looks, from outside, and B, does this work better? Yeah. B, what you might think about doing to explain yourself better to outsiders. It really does make a difference. And I point to the kind of thing that Hude mentioned at the beginning in genomics. People were, of course, interested in genomics, but people are very interested in cancer. And the association between those two provided a support for the field that carried it, for good or worse, in various directions. So what I'd like to do is talk a little bit about this issue of what one is explaining to whom. And then a bit about the question of function and structure, since they're really quite different and they're confused in this particular issue. A little bit more about explaining, and then some things about other factors which I think are important in healthy programs, if I can put it this way. So let me start by saying that I think listening to this morning has been intensely interesting for me. And it has very much the flavor of 1,000 flowers blooming. That is to say, obviously, a problem which we would all agree is intensely interesting and important. What is the brain? How does it think? What is mind? It fits right there with issues like what is life and where did life come from as being among the great problems of the next century. So I don't think you have to worry too much about that. The question, though, of whether people outside this field and outside this room can understand what it is that is going on and where it's going to lead is, I think, much more complicated. And I'll just make a point to you to set a starting point, and that is that when I first heard about 10 to the 14th neurons and synapses and the brain map and things of this kind, I've checked this with a bunch of people who are good scientists and actually neurobiologists, and everybody's opposed. I mean, almost uniformly. And the reason is not that it's a bad idea, it's just that there's a very deep skepticism that this approach, doing physical mapping, which is the way it's interpreted, at that scale is A, going to work, and B, going to lead to something. Now, I'm not going to argue with you whether that's correct or not, but I'm simply telling you that what you name this program is actually an important issue, because people do take the name for what it is. The Human Genome Project covered all sorts of activities in genomics, but it was an understandable concept. So what is the understandable concept here? And I think one of the issues for this group to think about, again, harks back to what Huda said, and that is that one of the very important functions that this group is doing, one of its privileges, is to begin to set the agenda for the field and to set the balance. What areas are going to be emphasized, what areas are going to be de-emphasized by assumption at the beginning. And that turns out to have a very big influence on the way the field works out over the course of time. So it's something to think about very carefully. Now, another point that's interesting, I have a very good friend, a man named Norm Augustine, who is as skilled a explainer, briefer, as anybody that I know. And it's very interesting to hear Norm talk about a problem of this sort. What he does when he discusses a new project is he spends 90% of the time in his discussion talking about what the problem is, and then 10% talking about the solution. And his argument is that if you can get the listener to agree to the problem, and agree that there is a problem, and agree that the problem is soluble, then the question of how you do it and the details are, in a sense, technical. But you have to get that basic agreement at the beginning that there's something there that's worthwhile studying. A very important issue, again, to think about here. Now, in doing that, of course, one has to project into the future, and there's a very delicate art I think, in thinking about programs that, of course, you can't predict the future, and of course it's reasonable to overpromise, but it's not a good idea to overpromise too much. Because if you overpromise too much, then what happens is that there's disappointment at some point, and that can be a bad idea. All right, a couple of things about function and structure. If one talks to outsiders about a geographical map, and the word map semantically is often considered to be locational. A functional map is one thing, but a map is geography. What connects to what? The general sense is, I don't care. I don't see that that necessarily has anything to do with how the brain or the mind functions. What you really care about is function. I mean, what's going on. A functional map could be a different thing altogether. One of the interesting things to hark back to is a really ponderous piece of scientific machinery called the scientific method. The scientific method is really ponderous, but it's served us pretty well in a variety of circumstances. And if you think about what it is this wheel that has the following characteristics. You make observations, you start with observations, you postulate an explanation for those observations, you test those postulates in terms of experiment, and then you go from that experiment to new postulates, and if you go round and round and round, you optimize your postulates, they become a theory. If you can find no exceptions to the theory, it becomes a law. Now, it has a very important characteristic to it, which, so far as I know, hasn't been mentioned so far, and that is the idea of falsifiability. That is, you have to be able to prove that something is wrong. And that issue is, it's easy to neglect in biology, and it's easy to neglect, I think, in many fields when they're just beginning, because you're collecting information, you're collecting lots of data. But the notion that if you get enough data, somehow truth will emerge from that, there aren't very many examples in which that's worked in science. So it's something to think about in the context of the kind of work that you're doing here. The second word that I want to bring up is information. And the brain, of course, in a certain sense, is an information processor. Now, how information turns out to be mind and behavior at the end is a complicated issue, which I certainly can't answer. But I think there's the assumption that there's something simple about information. And information is really very complicated, very complicated. The people like Bob Laughlin, who have periodically done this kind of thing for a living, have views of information which are very sophisticated, often on rather simple systems. But if you talk about information in the context of biology and ask, what is information in genomics? What is information in protein ligand interactions? What is the information content of the cell? How does information change? There actually isn't a theory of information in biology. And it's that kind of simple structural idea, entirely independent of issue of engineering models, where I think that there's a broad opportunity to balance the enormous enthusiasm for new technical methods with new conceptual guides to help to constrain this problem in a way that helps people to understand what to do with their data and what data to get, how to use the data to test hypotheses. Because we are ultimately interested in the information flow. Now there's information, there's networks, there's circuits, there's synapses, there are physical connections, there's behavior at the other end. How you put all that together, I think the field will figure out as it goes on, but it's not going to be a straightforward story to make. So, now how does one explain a field as complicated as this? And it's difficult. It's difficult whenever it works, and it's even more difficult when it doesn't work. I'm going to return to two examples in a bit which illustrate this. But I spend a lot of time trying to explain to various groups of people how you explain something. And I think there actually is a structure that you can think of, and it comes oddly enough from English, that is from story writing, and it is the idea of a story. And if you talk to people in English, they will tell you that the most elementary description of a story has only 4 components. The nice thing about 4 is anybody can remember 4. The 4 are, and think about this in the context of a murder mystery. You have to have a puzzle or a problem. Who killed the lady at the house? Was it the butler? Was it somebody else? So there has to be a conflict or a puzzle or a problem you want to resolve. The second element is what they call a journey or a trek, which is how you get there. And we're spending much of our time here talking about that part. What are the technical methods that you use to get data or to formulate experiments? The third component is that there has to be a surprise. So if you don't have a surprise, nobody's interested. You have to catch the attention of people. And you know, the surprise is, to say it's a theory of mind is probably too far off. What's something that's shorter term that people can actually get a grip on, believe, and work with, something that will catch their attention? And then finally, there's the resolution. The cat killed the lady of the house. It wasn't the butler. Or boy gets girl, or whatever it is that happens at the end. Often in science you'd call it an application. But if you don't have those components, you don't have much to work with when you're talking to people who are not neuroscientists. Now we are all, everybody is fascinated by the particular tack they take to their particular piece of research. But outside, it's a different story. People want to know what you're all doing, and it has to be simple enough that people can understand that. And it's very difficult to do. It's very, very difficult to do. So one of the issues here is to start hammering out that story. And genomics, you know, we're going to understand the genome, and basically it was, we're going to understand the genome. Based on the genome, we're going to understand cancer. Based on that understanding, we will cure cancer. And based on that, your mother will live for a longer period of time. It had those kinds of components. Now, it's turned out to be more complicated, as everything in biology is. But here, you've got an even more complicated problem. So how do you simplify this very complicated issues with top and bottom level stories in such a fashion that I, as an outsider, can understand what the field is going to be doing, what its deliverables will be? This is a very interesting kind of thing to think about. Now, in that context, there are just a couple of things to remember. One of them is the question of why now? And this problem has been around for a long time. And you know, this is hardly the first group that's thought about the nature of the brain. So why is now the time where we expect something astonishing to happen? With genomics, it happened because the technology of sequencing became so good that virtually anyone could generate floods of data and then begin to think about what could be done with that. What's the corresponding thing here? I don't know the answer to that. And then the second question, which is in the same general issue of why now, is who cares? And obviously you care because you're interested in the problems. But outside of this room, with people who are not neuroscientists, what do they care about? What is the problem that you are going to say that you are going to solve that people care about? And I don't think there's any shortage of these problems, all the way from alleviating tremor and Parkinson's disease to beginning to think about depression, which is one of the great problems in public health. I mean, you can think about all kinds of issues, but think about what that might be. Now let me just close with 4 other brief points for the group to think about. The first is this issue of deliverables. And I think that we heard about this earlier as well. It's really important to have deliverables and outcomes. They don't have to be things which have the characteristic that they are the ultimate goal, but you need milestones along the way so that you can go to the outside world with successes to show for what you've done. We have done this, it's a step along the way, but we've actually made progress and we've solved this problem. And our next problem is going to be something, and our next problem after that is going to be something. It's not a very compelling case to say that we're here, in 100 years we will have a theory of the mind, and there's nothing to show you in between now and then because it's all too complicated to understand. There has to be a series of steps. So what are those going to be, and what do they look like? A second issue is, I think, an important sort of scientific issue to think about, which is the question of reductionism versus higher level stuff. If you think that a theory of the mind is going to come by understanding the function of individual synapses and then building upward from there, that tends not to work too well with really complicated systems. It works well with engineered systems like transistors to integrated circuits to devices to the internet to Facebook. I mean, that's a system, but that's an engineered system. Picking really complicated systems apart is hard to do. So what often one does is to go from the end and look at higher level functions, behaviors, and see if you can explain those in terms of black boxes. And if you have good working models based on black boxes, you can begin to pick the black boxes apart. But just to give you an example of how the fully reductionist approach can run into difficulties, again, we can go to genomics. And if you talk now to the people in the pharmaceutical industry, what they will say is they're moving massively away from target-based medicine to phenotypic assays. That is to say, if you want to find out whether a mouse gets better, you give the mouse the stuff and you see what happens. You don't ask too many detailed questions. The detailed questions haven't worked out very well. So here I don't have a sense where the dividing line is between things that are best done at high level and things that are best done by going fully reductionist. But I think there's probably a place for everything, and zebrafish is obviously a nice transparent model, but it's probably not going to tell us very much about depression. People are probably more interested in depression than they are in zebrafish outside the room. That's an interesting question. A third point is the question of balance and inclusion. We are at the tail end of a pretty successful program in the United States on nanoscience or nanotechnology. The question of why was this successful, and that's complicated, but one of the reasons is that when this program emerged, it was phrased in such a way that virtually every area of science saw that there was something in it for them. That is, the chemists, the biologists, the physicists, the device guys, everybody saw that there was some value in nanoscience for them. And there was an supporting enormously important technology, which is the technology of integrated circuits. And what's happened over the course of time is what the engineers in Intel have done, which is almost beyond belief in terms of its sophistication. The 2 generations, or maybe 1 generation from now, microprocessor will have minimum feature sizes that are in the order of maybe 8 nanometers. I still can't believe this, using 190 nanometer light. But so they provided an enormous practical push for this area, and then everybody had something interesting to do at the nanoscale. The question is, how does one open this community in such a fashion that everybody thinks there's something interesting and important that they can contribute? And that's balance. It has to include engineering. It has to include clinical medicine. It has to include the molecular. It has to include cells and animals. I mean, the whole story has to be there somehow. But making the story inclusive will make a much stronger case for building a strong community and continuing on with it. And then the last point I will make is the conclusion of industry. Now, when you say inclusion of industry, this has a variety of meanings, but let me just tell you another final short story, which came from a component of genomics that was really very important, which was Illumina. And the Illumina sequencers have been as important as many other things in genomics. And the inventor of the technology at the very beginning for the Illumina system was David Walt, who's at Tufts, a very sensible guy. I know him fairly well since he was a postdoc with me at some point. And I was at a seminar with David in which one of the people in the audience at the end asked the following question. He said, how do you handle the conflict of interest problem in an academic laboratory when you're working on this and there's a company working on the same thing? How do you handle it? And he said, there's never a problem. And the reason there's never a problem is that once industry takes up an idea and good engineers, mature engineers begin to work on it, an academic laboratory can never compete. Now, the relevance to this is that if you think about what Illumina and other sequencers made possible in genomics, you can then ask the question is, are there corresponding things in this area where really skilled industrial engineers can make a capability available for the community in a way that makes it possible to collect all the data, the structure, the function, the measurements that you want to collect? Because it's going to be vastly, vastly easier if that's done as a centralized function with real people paying real dollars to get it done really, really well. And it may be premature to do it at this point. I don't know the answer to that. But it is something for you to think about. And I think that the earlier you get people who are professional engineers and on the other end of things, clinicians, actively involved in the work that you're doing, the more likely you are to find those components that you can use and those motivations for using them that will help to keep the field strong. So it's a fantastic area. It's unbelievably complicated. Outside, it looks much less straightforward than I think it looks to you inside, and inside it looks pretty chaotic. So you can imagine what it looks like outside. Thank you very much.
I think we're ready for lunch. So thank you all this morning for a very informative discussion. And this afternoon, we hopefully know where your assignments are. And the idea, just to reiterate, is to try to get to the point where some of the specifics can be written down so that we can converge to some recommendations eventually by tomorrow afternoon. Thank you.